Hi everyone, my name is Austin, and today I just want to share some really cool stuff with you. Maybe just sit back, relax, and enjoy my presentation. So, as a bio student, we all know that the oxygen is an essential part for metabolism, which refers to the chemical processes that occur within cells to convert food and oxygen into energy, as well as the synthesis and breakdown of molecules that are necessary for different cellular functions. And the primary energy generating process in cells is called aerobic respiration, and that is why sometimes aerobic respiration is also considered as a hallmark for eukaryotes. Well, however, there does exist in eukaryote that growing hypoxic environment, and the mitochondria of this organism has lost all or maybe parts of their genomes already. And this organism is today's focus. Some of you might already heard of it before in other courses, books, websites like online resources, and this is Henneguya salmonicola, the animal life without mitochondria. And just a side note, this is a parasite that lives in fish muscles and is also a species of myxozoan. So, as I said, this organism has no mitochondrial genome, and thus it has lost the ability to perform aerobic respiration. Well, but just hold up, just wait a second. A question may arise here. Well, like you may want to ask a question like, how does it stay alive, and what does its metabolic pathway look like? And exactly, how does it stay alive without being able to perform basic aerobic respiration? And I'm also curious to see how does the metabolic pathway work in this organism? And yes, that's also what the researchers were wondering as well. So they decided to do an experiment on this organism. And they first extracted DNA and RNA sequences in Henneguya, then analyzed them through various techniques, including electron and light microscopies, filtering, and assembling. Then they estimated the genome size. And finally, they were able to characterize its mitochondrial metabolic pathway with empty DNA and empty RNA. So here is the results. Figure A is from another myxozoan species, and it shows a large nuclei with many smaller mitochondrial nucleosomes. As you can see, the white arrows are pointing. The white arrows are pointing at the small pores, or the small circles. Yeah, those are the smaller mitochondrial nucleosomes. And on the other hand, figure B is from Henneguya salmonicola, which is showing a large nuclei, but surprisingly no mitochondrial nucleosomes, which you can see there's no such small circles or small nucleosomes as you saw in figure A. Hence, these results further prove that Henneguya salmonicola has no mitochondrial genes, which is kind of shocking. So what they decided to do is they further dived into the process of its metabolic pathway and to try to see how different its metabolic pathway is compared to a normal pathway. And they summarized their findings into two figures. So let's take a look at them. So figure A is the aerobic mitochondrion, like the normal ones, and figure B is the Henneguya MRO. Oh, sorry, by the way, MRO stands for mitochondria-related organelles. So figure A, this is the normal pathway of aerobic respiration in mitochondria. First, we have the DNA, RNA, and protein synthesis pathway. And on the left-hand side, we have the conversion from the pyruvate to acetyl-CoA, driven by pyruvate dehydrogenase complex, which oxidatively decarboxylates the pyruvate to form acetyl-CoA. Then the generated acetyl-CoA enters the citric acid cycle to further generate more energies with all the electron transport complexes. And in the graph, the TCA cycle is basically just another name for the citric acid cycle. So as we can see, there are a lot of electron transport complexes, but let's just pay close attention to two of them. The complex 3 and complex 4. So what complex 3 does is it accepts electrons from the electron carrier molecule, and complex 4 is the final protein complex in the electron transport chain. It receives electrons and transfers them to the final electron acceptor, which is the molecular oxygen. The movement of electrons through complex 3 and 4 releases energy that is used to pump protons across the inner mitochondrial membrane, creating a proton gradient, and this gradient drives the synthesis of ATP during oxidative phosphorylation. So essentially, Complex 3 and complex 4 are the most important electron transport complexes in this pathway. However, notice that Henneguya does not have these two complexes, well, and it doesn't have many other required molecules as well, which simply means oxidative phosphorylation cannot be carried on in Henneguya, and that implies it does not rely on aerobic respiration. So, the question now becomes, how does it obtain energy to not die? Well, the researchers further found out that one possible explanation is that it likely has adapted to survive in its host environment by relying on anaerobic metabolic pathways. So in its fish host, it just undergoes proliferation within the white muscles, which is known to do anaerobic metabolism, and then they can just obtain energy through that. The specific way of how it achieves that is still not known yet, and the reason why it evolves in a way like this is probably they always just live in the host, which is hypoxic environment, the fish muscles in this case. So the loss of the abilities to perform aerobic metabolism might be advantageous to them, since they always live in an anaerobic and hypoxic environment, which makes it seem not useful enough to keep that. And just one side note, the paper is published in 2020, and there wasn't a way to prove it's exactly how it obtains energy based on the research paper. And overall, the researchers did a really great job in understanding how the pathway looks like in Henneguya salmonicola. They provided us a detailed graph on how it looks like and how does it differ from normal ones. They also explained how it stays alive without the needs of oxygen and aerobic respiration, just like other eukaryotes. And at the end, they proposed the hypothesis of what evolves this way to lose the ability to produce energy from aerobic respiration. However, one thing I would actually love to research a bit more is, will Henneguya rely on other ways to obtain energy? So what I'm thinking is glycolysis and fermentation processes can be a reasonable hypothesis just without further involvement of aerobic respiration. And I think that would actually be an interesting thing to do more research about. Yeah, but still, this finding of Henneguya is still kind of shocking. If I recall correctly, this is probably the first eukaryote we found that does not require mitochondria to stay alive. And yeah, that's all I have for today. I hope you all learned something new, and I hope you all enjoyed the presentation. Any feedback would be appreciated, and thank you for listening. Sorry, I forgot to include a citation. So here's a citation, and thank you very much.